Hello, I'm David Brenner. I'm the National Plant Germplasm System Curator for quinoa in the United States, and I'm part of the National Plant Germplasm System, although I'm an employee of Iowa State University. The quinoa collection is in Ames, Iowa, associated with Iowa State University. And much of what I do with quinoa is grow plants to create new fresh seed stocks for long-term conservation, and I do that in a greenhouse. Today I'll give you a tour of our online GRIN database and show you a little bit about how to select and order germplasm for research, and then I'll finish up with a brief tour of one of our seed vaults with seed jars and seeds that are ready for distribution. So I'm going to toggle to GRIN now. This is GRIN, our publicly available database, and I'm going to emphasize that there's a new user button here where you would enter your name and address, and that would all be in our long-term records and would be a start toward ordering germplasm. And you do that the first time, and then on later visits you would just log in with this button. GRIN Global is the name of our database and system, and it's used in other countries also, but this is the United States Department of Agriculture. We distribute germplasm worldwide free of charge for research and development. So it's not for home gardening, but it's for research, and it's for developing new and better uses of crops to feed and help people, and I hope we can benefit you. And we're always trying to improve things a little bit, so the database keeps changing. We get more seeds available, we're adding more species, and trying to improve and change and have active curation here. I'm going to show you some of our features. There's a accessions button here, and I'm going to enter quinoa, and do a search. It takes a few moments for the list of accessions to come up, so it's working now. And here it shows about 10 accessions per page, and there's 399, so that's 399 that are available to distribute. And these on this page have Ames numbers, but most of them now have PI numbers. So the accession number usually has a picture, and I'll show you the picture. So this is how I see quinopodium accessions. I seldom grow them in the field because they're well adapted to growing in a greenhouse. These tents are about 6 square feet, and usually one tent is enough to get enough seeds for our purposes, and I can have often more than 50 of them growing at a time. I have usually 2 plantings per year in the greenhouse, one in August and one in January. And I'll click on Passport to show you things like who donated the germplasm to us, where it's from, and we also have this observations area where we enter notes we've taken about them, such as the panicle form, is it glomiform or amaranthiform. The 100 seed weight data is pretty good, so we have usually 100 seed weight data for each of our seed lots, so you get 3 approximations of what the seed weight would be. And we have male sterility data for most of the accessions. Now, male sterility is actually pretty rare, but we have good data about which accessions have male sterility observed or never observed. So it would be a good place to start if someone wanted to research further with male sterility in quinopodium. And I'll take you, so it's 10 accessions per page. I want to show you something on the 11th page, so I'm scrolling through them. And notice I've put pictures now of many of the leaves of many of the wild species. In the wild Chenopodium relatives, leaf shape is often diagnostic and difficult to describe, but I'm taking pictures of them as I grow them, and we're posting these pictures now. And here's page 11, where we have some University of New Hampshire donations, and here's the first Decker. And most of these Decker donations are of interest as for herbicide, either susceptibility or resistance. So it's a new specialty. We're collecting herbicide tolerant germplasm. And I'll take you back now to the descriptor part. So we just clicked on accessions. Now we're going to click on descriptors. And descriptors is usually large data sets taken across multiple accessions in a crop. And we have many crops all over the National Plant Germplasm System. Our station names is just one station, and other stations specialize in other things. So I'm scrolling through to quinoa. There's quinoa. And in quinoa, we have the descriptors that we've already been talking about some. And we have this crop detail page. So I'm going to click on crop detail page, and there's 4 columns. The descriptors you've heard a little bit about, the species, which is taxonomic information, and I'll show you citations in a minute. So species, these are the species that we include within the quinoa group, and they aren't all quinoa, and they aren't all even Chenopodiums. They're related species that are administratively grouped this way. So Blightum natalianum is mostly Great Basin, United States. It has a beet-like rounded root that's a traditional Native American food, and edible seeds, and edible spinach-like leaves. Chenopodiastrum is almost like a Chenopodium, but there are little differences that make it in a different genus. And these Dyspanias are aromatic. They include some spice. Some use as spice. Quite a few things, but enough like quinoa that they're classified together. Cycloma is one of my favorites. It has large seeds, and it's traditional quinoa-like food in the Great Basin of the United States, and the plants break off from the ground and roll about as tumbleweed. We have 3 accessions. Nebraska, one I collected recently in Iowa. And this is the same format we've seen before at other parts that I've shown you, but I'll click on more of the columns this time. This, if you were going to place an order, you could click here and the accession would be added to your shopping cart. And we're not going to order today, but we could. And I'm going to click on NC7. NC7 is a code name for the particular station here in Ames, Iowa, where I work. And we have many crops here, vegetable crops such as carrots. Maize is the big crop. We also have sunflowers, woody landscape plants. And I'm just one of the curators, but I have many crops working here, including forage legumes, spinach, parsley, dill, fennel, amaranth, and many other crops. And if you clicked on the species name, you'd get pretty much the same format, but a little bit more background. So I'm going to go to citations now. So I'm going back to, this is the species list. Oh, here's quinoa, I think, which is most of you are interested in. 145 are available. And Ficifolium is one of the species that's an approximation of half the genome of quinoa. So hypothetically, you could use Ficifolium and another species with the other genome to make a synthetic quinoa. We have only 6 accessions. And I'll show you this one that I think is kind of fun. Occasionally I segregate new accessions from old accessions. So I was growing this one and I found this stocky dwarf mutant type and segregated it as a new accession. And it looks like this, with these rounded leaves. And it was collected under a bridge in Tampa, Florida. And I'll show you a standard type, Ficifolium. This is one donated by the research group in New Hampshire. And it has a much longer leaf shape. So, as I said, I was going to take you to the next feature, which would be citations. Let's go there now. This is the list of species. Berlandii is the closest approximation of quinoa, and we've got most of the subspecies of Berlandii. And so somehow I took you back one too far and got out of the quinoa crop group. There, now we're back in quinoa, and we'll go to the citations. Citations allows us to enter bibliographic citations for references that cite our germplasm. And I load them here, and it gives usually the DOI, which would take you right to the publisher's information about the paper. It doesn't get you past the paywall to get the paper for free if there's a charge, but it takes you to usually the abstract. And some of us are affiliated with university libraries, and we can get in for free from there. And it cites links to the accessions that are cited there. So I'm going to click on this 7 accessions that are cited by that particular paper, and this makes it easy for you to order the same accessions that a previous researcher used for maybe similar research. Here's a studies the genome. Fogg worked with domesticated chenopodiums in Taiwan, and lots of great papers. Things from India. Some of our authors have, especially in Pakistan, there's some Pakistan papers here, like Munir's, where they very successfully introduced quinoa from our germplasm as a crop for Pakistan, and these publications are here. So that's most of what I wanted to show you, and I'd like to emphasize that we're here to help. We really do send germplasm worldwide free of charge for development projects, and I hope that good things will come of it. The system keeps changing on our side. We add more accessions, and we are sometimes running out of seeds and then growing more seeds to make them available again, so different accessions can be unavailable and then available other times. Keep checking back. And the GRIN database itself changes. It's gone through dramatic changes in the last couple of years. It'll keep changing. And that's most of what I have, but from this point in the talk, we'll toggle to a little tour of our seed vault. So thank you very much.